I'm Suet-Feung Chin. I'm Senior Associate Scientist with the Caldas Group, and we are based in the Cancer Research UK Cambridge Institute. Cambridge Institute is an institute based in University of Cambridge. We do translational research, and we are fully funded by CRUK. The lab is a functional genomics lab. We work basically on breast cancers. We use genomic methods to identify to molecularly characterize breast cancer so that we can understand breast cancer. And we've so far identified 11 subtypes of breast cancer. And we would like to expand what we've done previously on 2,000 breast cancers to 10,000 breast cancers using FFPE material. For prospective studies, most of our samples are fresh frozen tissues or blood. We also have plasma. For retrospective studies, they are all formalin-embedded paraffin material. Before, we use column-based methods, which were all manual. So the challenges would be very hands-on, very time-consuming to do the extractions. And when you have to do a lot of samples, it's very painful. So the Promega Maxprep and Maxwell combo allowed us to do 48 samples each run. And it would take about 90 minutes on the Maxprep for the aliquot of all the samples. Essentially, it's all automated. We only have to barcode the samples in, put it onto the Maxprep, press go. And then once it's done, we transfer the tray and barcode the tray into the Maxwell system, which would share the same LIMS system, and all the samples will be in the Maxwell. We press run on the Maxwell. It takes about 45 minutes for the 48 samples to be done. And once the DNA is extracted, we transfer the tray back onto the Maxprep, where we barcode the tray in. Again, the samples are recorded. And then we do the post-processing method, where we transfer the eluted DNA into the 96-well plate. So it's essentially really fast, and it's hands-off for most of the protocol. The pre-processing methods on the Maxprep is very easy to use. There are some items that can be customized, like the number of samples and the amount of elution buffer, but other than that, it's plug and play. It's really, really simple to use. The Maxprep provides post-processing methods for sample transfer, for normalisation, for setting up PCRs, and for quantification. The one that we consistently use is the sample transfer. The Maxprep interface was really easy to use. It's self-explanatory, except for a couple of customized items that we can change, such as the number of samples that we run and the amount of elution buffer that we require. Everything else is pretty straightforward. We routinely use the Maxwell FFPE kit because of the nature of the project, but we've also used the cell culture kit to extract DNA from cells. We've used the blood kit to extract DNA from blood, and we've also used the Maxwell RNA kit. What I like about the Maxwell FFPE kit is that it uses mineral oil to dewax the sample, and using mineral oil without having any additional wash steps, like when you use xylene and ethanol, means that you lose less sample. The DNA yield from the Maxwell FFPE kit has been comparable or even more than the existing manual methods that we've used, and the quality has been really good. We've extracted more than 5,000 samples, and we've measured the OD of about 800, and they're all always between 1.8 to 2. Each run that we do on the Maxprep takes about 60 minutes for 48 samples. And the extraction of 48 samples on the Maxwell will take about 40 minutes. And then the sample transfer, post-processing on the Maxprep will take about 10 minutes. Compared to the other manual method, which would take about 3 hours, and is hands-on all the time for 3 hours. The Maxprep and Maxwell has made the extraction method very consistent and very robust. We get very good DNA yield and quality from all the samples that we've done so far. I like the Maxprep system because it's fully automated and it's plug and play. We don't really need to change anything. It's all pre-programmed, and it saves us a lot of time, a lot of hands-on time. This project will involve 10,000 breast cancers. It will expand what we already know about breast cancers in 2,000 samples or more. And it will allow us to understand heterogeneity and what are the different drivers of breast cancer to allow us to better understand the disease and be able to help the patients.